All right, so in this video, we're going to look at translation. So now we've taken our RNA, we've transcribed it in the nucleus from our DNA piece, and we've processed it. So we cut out all the introns, remain the exons. We have a 5' cap and we have a poly A tail. And so what's going to happen now is there's three kind of main events. So the first event is the initiation event. And you can see that you have a little piece of mRNA, and this is the small subunit of the ribosome. So in this case, the mRNA binds to the small ribosomal subunit. Okay, and that's so that we can like draw a little line there. And then what's going to happen then is you're going to have this tRNA come in. And this is a special tRNA. This is actually the very first tRNA. And if you notice, its code is UAC, and it always binds with AUG. AUG is the start codon. So that's kind of, so you might want to highlight that. So that AUG. And so the initiator, tRNA, base pairs with the first codon on that mRNA, on the mRNA. And so you notice it's AUG, and so the A is going to bond with the U, and the A here is going to bond with this U, and then this G is going to bond with this C. Okay, so they're going to base pair like that. And the first amino acid always, 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 for whatever reason, is methionine. And so that's where that first, so it always starts with the AUG. That's always the start codon. Okay. Then at this point, what's going to happen is the last stage of the initiation sequence is we're going to have the large ribosomal subunit. So that's this part here. Okay. So the large ribosomal subunit will bind to the complex. And what's happening now is there's actually a couple sites here. So there's two different sites or spots. This spot right here where this initiator one is, it's actually at the P site. So I'm going to kind of draw a line and write P. And then this site's at the A site. And they stand for peptidyl tRNA transfer or binding site and amino acetyl tRNA binding site. Just no P and A. Okay. And so right now the initiator tRNA, or that one that has the UAC with the methionine, is located at the P site. Okay. Now what's going to happen is we're going to try and elongate our polypeptide or our protein. And so in the elongation stage, the first stage here is you'll notice that the next codon, so this one says GCU, and so then what's going to happen is CGA is going to come in. So that second tRNA with its amino acid attached is going to base pair with the second codon on the mRNA at the A site. So it comes into the A site. Okay. And then what's going to happen is, so we have this one coming in, we're going to start to form a bond there. There's actually an enzyme that does it, and it's the only enzyme you have to know in this whole thing, and it's got a terrible name, and I'm sorry. Okay, well, it's got an okay name. So peptidyl, and you can't actually see it in here, which I know is annoying. It's actually inside the ribosome. So peptidyl transferase is the name of this enzyme that we have to know. It's going to form a peptide bond between the first and second amino acid. Okay, so it's going to form that peptide bond right there. Then what's going to happen is, after that happens, the first tRNA will exit. That's what's happening here. The mRNA is going to shift by one position. So now the second tRNA is at the P site, and the third tRNA is going to come in and base pair with the third codon. So the third tRNA will then base pair with the third codon at the A site. And then it just continues until there's a stop codon that comes into this A site. So it's just going to keep shifting, and then peptidyltransferase will make this peptide bond, and then it'll shift, and this one will fall off, and a new one will come in, and they're just going to base pair like that. Okay. Now, that's the last and final stage, is this termination stage. And in this stage, what's going to happen is this codon here is going to be, and it'll be much longer than a 3 thing. This will end up being a stop codon. And so there's actually 3 stop codons. There's only one start, but there's 3 stops. So now a stop codon comes in. And so there's 3 stop codons, which is, and they're very similar in sequence, UAG, UAA, and UAG. It's going to come in, and so it's going to reach the A site. And so then in little molecule binds, and this molecule has the easiest name ever to remember. It's called release factor, and it's literally going to release everything. So it's going to bind to the stop codon. There are no tRNAs that bind that have the anticodon for these. Okay, so there's no tRNAs that have that happen. And then this polypeptide is going to be released. So my polypeptide will be released. And then there's all kinds of molecules that are going to help to, there's little chaperonin proteins that will help for that polypeptide chain to fold. And if this ribosome is stuck on the ER, like on the rough ER, it's going to help package and ship that somewhere, and it could go to the Golgi. There's all kinds of things that can happen there. And your book kind of talks about them, and we'll talk about them when we talk about regulation and stuff like that. But basically, it's going to be released. Okay, so it's hybridized from this tRNA at the P site, so we're going to let it go. See you later. And then really what happens is, in kind of the final step, is this whole thing just breaks apart. And that's kind of, so this whole complex just disassembles. Now, the cool part is all the pieces can be used again. Okay, so this mRNA can actually be used again. It can be used again to make another protein, the exact same protein. And the same with these ribosomal subunits. They can rebind to this particular mRNA or a different mRNA. Kind of just depends on what's going on in the cell and what the molecules of the cell are telling this mRNA to do and these ribosomal subunits to do. But you can use that same mRNA over and over and over again to make the same thing over and over and over again. So that's kind of translation. That's exactly how a ribosome can take an mRNA and translate it into the language of a protein. And so that's kind of how that works.